Hello everyone, my name is Donghoon Lee and I'm from Mount Sinai, Department of Genetics and Genomic Sciences. I want to quickly thank the organizers for giving me the opportunity to present my work here at the ENCODE Consortium meeting. The work I'm going to present to you today is about building a comprehensive list of human enhancers using whole genome STARR-seq assay. As we all know, non-coding regions harbor many mutations linked to complex human diseases, and we do not yet have a comprehensive list of cell-type specific enhancers. Identifying and understanding the function of enhancers has long been a challenging objective of the ENCODE Consortium. The original STARR-seq has been proposed by Stark et al. In 2013, and there are several advantages of using STARR-seq over many other enhancer assays. First of which is that it can be used to unbiasedly and quantitatively measure the enhancer activity. Also, it can be scaled to the whole genomic level. In collaboration with White Lab, we have scaled the original replication-based STARR-seq to whole human genome level, and we generated the STARR-seq datasets from 6 different ENCODE cell lines. In comparison to the fly genome, which was used in the original STARR-seq study, the human genome is about 25 times the size of fly genome, and this alone makes it very challenging to scale the STARR-seq libraries to comprehensively cover the whole human genome. In addition, to help identify true applications from PCR duplicates, we split the cDNA libraries into 160 sub-reactions, each with a unique UMI barcodes, and we pulled them for sequencing. These advancements made the downstream processing more complex and challenging, so we carefully thought about a way to uniformly processing these enhancers while addressing any potential biases and artifacts from the assay. Here we have developed an end-to-end processing pipeline for STARR-seq assay called STARRPeaker, and I'm going to explain several advancements over the previous method in the next few slides. STARRPeaker first uses the paired end reads to infer the size of the fragments, and uses the center of the fragments used for calculating the enrichments. In our analysis, compared to the read-based depth calculations, fragment-based coverage provided much sharper and better enrichment signals. Figure on the right side shows this point. Basically, we find smaller peak size and much higher peak fold enrichment, and also the negative log p-value. Next, we built more precise background model for accounting over dispersion in the STARR-seq coverage. In comparison to Poisson and binomial distributions, we found that the negative binomial distribution offers more precise fitting when we simulated these coverages. We also included the several sequence features as a covariates to adjust for any potential source of biases in the STARR-seq assay. More specifically, we used the DNA input coverage, GC content mappability, and RNA thermodynamic stability of the tested sequences. When we plotted the STARR-seq output over these sequence features, we observed a strong correlation indicating that STARR-seq may have been suffering from these systemic biases, and we need to adjust for these when calling on active elements. Moreover, unlike ChIP-seq, where both input and outputs are from the same DNA origin, STARR-seq measures the regulatory activity based on the abundance of transcribed RNA. Single-stranded RNA starts to fold upon transcription. Therefore, we suspect the resulting RNA structure might have an influence on the STARR-seq readout. As shown on the figure right, we saw a negative correlation with the folding energy, indicating that the more stable structures tend to have a higher STARR-seq readout. When we compare the enhancer identified by STARRPeaker to the previous method, we saw that STARRPeaker peaks have much improved the epigenetic enrichments in terms of DHS and enhancer marks. Also, we saw a higher number of TF binding events in the peaks found by the STARRPeaker. We also made a comparison to the public dataset to see how the STARRPeaker peaks compared to others. Here we show the STARRPeaker peaks had the best overlap among the other methods that are published by other people. Finally, we made a comparison to previously characterized enhancers by CRISPR-I tiling deletion. Here we show that although all 3 methods have successfully found the MYC enhancers, we do notice that other methods also find significant number of potentially false positive peaks, making the STARRPeaker a more specific method of all. This is end of my presentation, and of course this has been a collaborative effort between the Garcin Lab and the White Lab, And I want to thank both mentors, Mark and Kevin, as well as the researchers, Yoshi, Manman, Jennifer, and Martha, for collaborating on these efforts. Thanks for listening.